Welcome to the class, everyone. Topic for the day is somatic embryogenesis and artificial seed production. I am Dr. Shalini Singh from the Department of Biotechnology. In vitro somatic embryogenesis, first of all, demonstrated in 1958 by Stewart and Reynolds. There are two ways by which somatic embryos could be obtained. First, indirect somatic embryogenesis, and second way is direct somatic embryogenesis. In case of indirect somatic embryogenesis, the callusing is induced from the egg plant by rapid cell division, and then this callus will give rise to somatic embryos. Whereas in case of direct somatic embryogenesis, somatic embryos are developed directly from the egg plant without an intermediate callus phase. In dicotyledonous plant, somatic embryos passes through the globular, heart, torpedo, and cotyledonary stage. So these are the four developmental stages of embryo. One is globular, second is heart, third is torpedo, and fourth one is cotyledonary stage. It is similar as happens in case of zygotic embryogenesis after the fertilization process. The only major difference is that somatic embryos do not pass through the desiccation and dormancy phases as the zygotic embryo undergoes, but rather continue to participate in the germination process. This is a globular embryo, this is early heart shape, this is late heart shape, this is torpedo shape, this is early dicot, and this is fully developed dicot embryo stage. Steps involved in somatic embryogenesis are three steps. First is induction of embryo, second is embryo development, and third is embryo maturation. If you compare organogenesis and embryogenesis, what happens here? Embryo is a bipolar structure rather than a monopolar one. The embryo arises from a single cell and has no vascular connection with maternal callus tissue or the cultured egg plant. During organogenesis, shoot or roots develop from a group of cells, resulting into chimera formation, which later establish a strong connection with the maternal tissues. That is the difference here. Second, induction of somatic embryogenesis requires a single hormonal signal to induce a bipolar structure which is capable of forming a complete plant. Wherever in organogenesis, as you know, it requires two different hormonal signals to induce shoot first and then root organ. What are the factors which affect the success of somatic embryogenesis? These are the genotype and type of egg plant, growth regulators, and nitrogen source. Genotype and type of egg plant, first factor affecting the success of somatic embryogenesis. So somatic embryogenesis is also genotype dependent. For a given species, and significant variations in response between cultivars have been observed in several plants like wheat, barley, soybean, rice, alfalfa, etc. Genotypic variations could be due to endogenous level of hormones. Therefore, if the species has not shown somatic embryogenesis previously, then it is required to test number of different cultivars of that species. What tissue should be used in an explant in a particular species to induce somatic embryogenesis? The explant selection is much more important than the media selection for somatic embryogenesis process. The success of your experiment mostly depend upon the explant which you are selecting for your experiment first, and then you will go for selection of media. So you can take immature zygotic embryos. The best explant is cells that are already in embryonic state. In Azardica and Indica, that is basically commonly known as neem, the globular embryos did not show any response. The older embryos germinated, formed calli or differentiated three types of organized structures like shoots, somatic embryos, and neomorphs. Neomorphs are abnormal or embryo-like structure with different morphology. Often the same explant differentiated more than one kind of regenerins. The most responsive stage of embryos was early dicotyledonous, followed by torpedo-shaped embryos. Growth regulators, which you are going to use in the process of somatic embryogenesis, may be auxin. Auxin play a major role in the development of somatic embryos. Require a synthetic auxin for the induction of somatic embryos, followed by transfer to an auxin-free medium for embryo differentiation. 2,4-D, that is 2,4-dichlorophenolic acid, is the most commonly used auxin for the somatic embryogenesis induction. Other auxins, including norethylene acetic acid, NKA, indole butyric acid, IBA, picloram, that is 4-amino-356-trichloro-2-pyridine carboxylic acid, and indole acetic acid, that is IAA. A naturally occurring auxin, indole acetic acid, is a weak auxin and more readily broken down compared to 2,4-D and NKA. The auxins, particularly 2,4-D, is used in the concentration range of 0.5 to 1 mg per liter. Localized groups of meristematic cells in the callus, which are known as pro-embryogenic mast cells, that is PEMs. These are cell clusters within cell population which are competent to form somatic embryos. They are put onto proliferation or induction medium and stimulate the formation of embryogenic callus with PEMs, which have been indicated here by arrows in the induction medium, that is callus induction medium. In repeated subcultures on the proliferation medium, the embryogenic cells continue to multiply without the appearance of embryos. However, if the PEM, the embryogenic cells, are transferred to a medium with a very low level of auxin, that is 0.01 to zero 0.1 mg per liter, or no auxin in the medium, which is known as embryo development medium, they will develop into embryos. The presence of an auxin in the proliferation medium seems essential for the tissue to develop embryos in the embryo development medium. The tissues maintained continuously in auxin-free medium would not form embryos. Therefore, the proliferation medium is called the induction medium for somatic embryos, and each PEM is also known as an unorganized embryo. Cytokinins. Only few reports of somatic embryo induction and development in cytokinin-containing medium. Cytokinin in general induce somatic embryos directly without the callousing of explant. In most cases, TDZ, thiazolone, is used as cytokinin. This is a herbicide which mimics both auxin and cytokinin effects on growth and differentiation. The other cytokinins are also used in zygotic embryos, are used as the explant source. The most commonly used cytokinins are BAP and GA10. Cytokinin also result in the formation of somatic embryos as well as neomorphs. Neomorphs are suppressed embryos with green, smooth, shiny surface and solid interior, although they were epidermal in origin like somatic embryos with heart-shaped notch but showed monopolar germination, and there was no clear-cut reticular region in that. Nitrogen source. The most important nutrient of the culture medium is nitrogen, which affects the somatic embryos significantly. Form of nitrogen have a strong influence on the induction of somatic embryos. Often, the presence of ammonium or some other source of reduced nitrogen is required, such as glycine, glutamate, or casein hydrolysate. The ratio of ammonium to nitrate has also been shown to affect somatic embryogenesis. In few cases, the alkali initiated on a medium with KNO3, that is potassium nitrate, as the sole source of nitrogen and failed to form embryos upon removal of oxygen. However, the addition of a small amount, around 5 millimolar, of nitrogen in the form of ammonium chloride in the presence of 55 millimolar potassium nitrate allowed embryo development. Embryo maturation and germination. Germination of somatic embryos can occur only when it is mature enough to have functional shoot and root effects which are capable of meristematic growth. Somatic embryos show poor germination quality with respect to their convertibility into complete plantlets. This is because these embryos do not go through embryo maturation phase, which is a characteristic of seed or zygotic embryos. During this phase, accumulation of embryo-specific reserve food material and proteins impart desiccation tolerance to seed embryo and thereby promote their normal development for germination. Abscisic acid, that is ABA, prevents precocious germination and promotes normal development of embryos and is reported to promote embryo maturation in several species. A number of other factors like temperature, shock, osmotic stress, nutrient deprivation, and high density inoculums can substitute for ABA, presumably by inducing the embryos to synthesize a hormone. ABA is known to trigger the expression of genes which normally express during the drying down phase of seeds. Probably the product of these genes impart desiccation tolerance to the embryo. Secondary somatic embryogenesis is a process in which new somatic embryos are proliferated, which are originally formed on primary somatic embryos. Secondary somatic embryogenesis has some advantage over primary somatic embryogenesis, such as high multiplication rate, long-term repeatability, and independency of an explant source. Secondary somatic embryogenesis also overcomes post-fertilization barriers of the embryo. Immature embryos of interspecific plants from incompatible crosses may be reduced by culturing them for secondary somatic embryogenesis. It can also be used for the production of somatic embryos of species in which the embryos are the reservoir of important secondary metabolites. So, in such cases, secondary somatic embryogenesis can be practiced. Synchronization of embryo development. Generally, the differentiation of somatic embryos in semi-solid medium or liquid medium is highly asynchronous, which adversely affects the germination rate. Synchronization of embryo development is very important for artificial seed technology. Of the several approaches tried to achieve this, the most effective method are the physical separation of embryogenic stages and use of growth regulators to physiologically synchronize the development. The other alternative methods are the fractionation of embryos of different stages by filtration of suspension through mash of different sizes or by gradient centrifugation. Besides, the most effective method to achieve synchronous development of somatic embryos is the use of substances that would induce reversible cessation of embryo development at a particular stage. ABA at low concentration is the most satisfactory substance for this purpose. For example, in carrot, it inhibits the growth of roots and enhances suspension with torpedo-shaped embryos. Production of synthetic seeds. Synthetic seeds are also known as artificial seeds. It is possible to use naked embryos for large-scale planting, but it would be beneficial to convert them into synthetic seeds or syn seeds for large-scale clonal propagation at commercial level. This can be achieved by encapsulating the viable somatic embryos in a protective covering. The coating material should have several qualities. First, it should be non-damaging to the developing embryos, mild enough to protect the embryos and allow germination, but it must be sufficiently durable for rough handling during manufacturing, storage, transplantation, and planting. Contain nutrients, growth regulators, and other components which are necessary for germination. Quality of somatic embryos should be good enough. They all are of uniform stage with reversible arrested growth and with high rate of conversion to plantlets. There are two types of synthetic seeds. First are desiccated synthetic seeds and second are hydrated synthetic seeds. Desiccated synthetic seeds involve encapsulation of somatic embryos followed by their desiccation and can be prepared by first mix equal volumes of embryo suspension and polyox. Then the suspension was dispensed on a Teflon sheet which have dried suspension sticks to glass plate. Dried wafers in a laminar airflow hood for about 5 hours until the wafers get separated from Teflon plate. The polyox is readily soluble in water and dries to thin film. It does not support the growth of microorganisms and is non-toxic to the embryos. Hydrated synthetic seeds. Several methods have been examined to produce hydrated artificial seeds, of which calcium alginate encapsulation has been the most widely used. It can be prepared by following steps. First, mix somatic embryos with sodium alginate. Then, drop the mixture using a pipette into a 100 millimolar solution of calcium nitrate. An exchange reaction occurs and sodium ions are replaced by calcium ions, forming calcium alginate seeds. Coming to the applications of somatic embryogenesis, to speed up the clonal propagation of plants being polar in nature, being single cell in origin, there is the possibility to automate large-scale production of embryos in bioreactors and their field planting as somatic seeds. Bipolar nature of embryos allow their direct development into complete plantlet without the need of a rooting stage as required for plant regeneration in organogenesis. Epidermal single cells origin of embryos favor the use of this process for plant transformation. It can be used for the production of secondary metabolites in species where embryos are a reservoir of different biochemical compounds, efficient transport and storage, and to overcome embryo abortion, seed dormancy, and self-sterility and plant. What are the limitations of somatic embryogenesis? Complete conversion into plantlets or poor germination of embryos. Compared to other plant species, active research on somatic embryogenesis involving forest trees has been very low. The lack of knowledge controlling somatic embryogenesis, the synchrony of somatic embryo development, and the low frequency of true to type embryonic efficiency are responsible for its reduced commercial application. To obtain a complete conversion into plants, it is necessary to provide optimum nutritive and environmental conditions. That was all about basic information in somatic embryogenesis and artificial seeds. If you have any question, you can always ask me via message or mail. Thank you so much for listening carefully.